Hello my dear students, welcome back to Biology at Fyojit. Imagine observing a tissue sample under a microscope without any stain. What would you see? Almost nothing. Most biological tissues are transparent, but without staining, identifying nuclei, cytoplasm, chromosomes or connective tissues become extremely difficult. So how do histologists or pathologists and other biological researchers make these microscopic structures visible? The answer is biological dyes and stains. Today we will discuss introduction to biological staining, importance of staining, classification of dyes, important properties of stains, detailed discussion on Haematoxylin, Eosin, Carmine and Orcein, comparative differences and applications. This lecture is highly useful for undergraduate, postgraduate, life science, zoology, botany, biotechnology, microbiology, medical science and allied health science students. Let's begin. Introduction to biological staining. Biological staining is basically a microscopic technique used to enhance the contrast of cells, tissues and cellular components. Most biological specimens are naturally transparent, therefore stains help differentiate structures based on their chemical composition and affinity for dyes. Now the students remember an important distinction, that a dye is a colored chemical substance capable of imparting color to tissues, whereas a stain refers to the practical application of dyes during microscopic examination. So in simple words, dye means coloring substance and stain means process of applying dye. Importance of biological staining. What is staining? Why is staining important? Biological staining helps to enhance visibility of cells and tissues, differentiate cellular organelles, diagnose disease, study tissue organization in histology, perform cytological investigations, identify microorganisms, detect abnormalities including cancers. Therefore, staining is indispensable in histology, pathology, microbiology and medical diagnostics. Classification of biological dyes. Biological dyes can be classified in several ways. At number based on chemical nature or charge, basic or cationic dye, that's the first category. This possesses positively charged chromophores. They stain negatively charged acidic structures such as DNA, RNA, nucleus, ribosomes, etc. Examples include methylene blue, crystal violet, basic fuchsin, etc. Remember, basic dyes stain acidic structures. Acidic or anionic dyes, these possess negatively charged chromophores. They stain positively charged components such as cytoplasm, muscle fibers, collagen fibers, etc. Examples include eosin, acidic fuchsin, orange G, etc. Third category is the neutral dyes. Neutral dyes contain both acidic and basic components. Examples include Romanowski stain, Wright stain, etc. and they are mainly used for blood smear staining. Classification based on origin. Natural dyes obtained from plants, insects or lichens. Examples Haematoxylin, Carmine, Orcein, etc. And synthetic dyes, they are prepared chemically. Examples include Eosin, methylene blue, etc. Third category of the dye, third classification, which is based on staining method. Progressive stain, the stains in which tissue is left in the dye until the desired intensity is achieved without removing excess stain. Regressive stains, stains in which tissue is deliberately overstained first and then excess stain is removed or differentiated to obtain the required contrast. Direct stain, stains that bind directly to tissue components without requiring a mordant or intermediary substance. Mordant stain, what is mordant stain? Stains that require a mordant such as alum or iron salt to form a dye tissue complex and improve staining affinity. Ideal properties of biological dyes: it has highly tissue affinity, it has stable during storage, it is showing uniform staining ability, it has distinct coloration, it has solubility, it is resistant to fading, it has specificity toward target structures, and it has low toxicity. Haematoxylin. Now let's discuss one of the most important stain, that is haematoxylin, which is a natural stain widely used for nuclear staining within cell. It is obtained from the heartwood of Haematoxylin campesianum. The common name is logwood tree. It is native to Central America and Mexico. Important concept is the haematoxylin itself is not the active staining agent. After oxidation, haematoxylin is converted into hematine. Hematine acts as the actual staining compound. Classification: haematoxylin is a natural dye. It's a basic stain with mordant. It is nuclear stain. Properties: it requires oxidation. It requires mordant such as alum or iron salts. It produces permanent staining. It has strong affinity toward nucleic acids. Mechanism: hematine and mordant forms a dimetal complex which binds to DNA, chromatin and nucleus. Color produced: nucleus is either blue or purple, chromatin becomes dark blue. Uses: it is used in histology, haematoxylin eosin staining, pathology, cancer diagnosis and histopathology. Advantages include: it is excellent nuclear detail and permanent staining. Eosin. Eosin is a synthetic acidic dye. It is prepared synthetically from fluorescein derivatives. It is basically three types: synthetic dye or acidic stain and cytoplasmic stain. It is negatively charged, stains positively charged protein and produces pink or red color. Within cells, cytoplasm, within muscle fibers, or within our body, extracellular matrix component, collagen fibers, RBCs are stained by the eosin. It makes the cytoplasm pink, muscle fiber becomes red in presence of eosin, and collagen become also pink. So eosin is used as a counter stain in haematoxylin eosin staining. It helps to differentiate cytoplasm and connective tissue components. Carmine. Carmine is a natural red dye obtained from dried female coccineal insects, the scientific name of which is Dactylopius coccus, the active component of which is carminic acid. Its main target is chromosome, nucleus and glycogen. Acetocarmine staining is used for mitosis study, meiosis study and chromosome analysis. Thus carmine is highly important in cytogenetics. Orcein. It is derived from lichen such as Roxella tinctoria. It is of three types: natural dye, nuclear stain and elastic fiber stain. It is basically two types: acetoorcin, which is used for chromosome staining, and orcin elastic stain, which is used for elastic fiber staining. Chromosome, elastic fibers and nuclear materials are stained by this stain, by this chemical orcin. It is useful in cytology, histology and diagnosis for elastic tissue disorders. Comparative differences: haematoxylin is stored source is logwood tree. It target nucleus. Color is blue. Used in histology. Eosin source, it is synthetic. It targets cytoplasm. Its color is pink. Used in counter staining of within haematoxylin eosin. Carmine is the source is insects. Target chromosomes. Its color is red. Used in cytogenetics. Orcin, its source is lichen. Its target is chromosomes and elastic fibers. It is purple red in color and it is used in chromosome studies. Differences between haematoxylin and eosin: haematoxylin stains nucleus blue and eosin stains cytoplasm pink. Together they form haematoxylin eosin staining, the most widely used staining method in histopathology worldwide. Applications of biological stain: histopathology, medical diagnosis, cancer detection, cytogenetics, embryology, microbiology, forensic biology, tissue engineering research. Conclusion: to summarize, biological stains are essential tools in microscopy. Haematoxylin stains nuclei, eosin stains cytoplasm, carmine is important for chromosome studies, orcin stains chromosomes and elastic fibers. Understanding these dyes is fundamental for students of life sciences and medical sciences. If you found this lecture helpful, please like, share and subscribe to Biology at Piyojit for more detailed classes on histology, cytology, cell biology, evolution, human physiology, etc. See you in the next class. Stay curious and keep learning biology. Thank you.